Hello everyone. In this particular video lecture, we will be discussing about the microbial metabolism, its introduction and the energetic concepts. So now, as you all are aware with the structure of what exactly the prokaryotic cells are, we can go ahead with the discussion of the activities or the metabolic pathways, or we can say the biochemical reactions that enables these organisms to thrive. So, the life processes or the biochemical reaction of even the most structurally simpler organisms, that is the microbes, they also involve a large number of complex biochemical reactions. So, most, not although all, of the biochemical processes of the bacteria, they also occur in the eukaryotic microorganisms or the eukaryotic cells. That is, or as well as in the multicellular organisms, including humans. However, there are some of the biochemical pathways or the microbial reactions that are so unique to bacteria, and these are very fascinating. Why? Because they allow the microorganisms to do the things that the eukaryotes or the humans or the plants cannot do on their own. For example, some of the bacteria are able to thrive on cellulose, whereas some others can live on petroleum as well. We all are aware of the fact that plants cannot fix nitrogen on their own from the atmosphere, and therefore, this nitrogen fixing process or the availability of nitrogen, making the nitrogen or the atmospheric nitrogen, gaseous nitrogen, available to the plants is done by the Azotobacter species. So these were the some examples of the biochemical reactions that makes the microorganisms unique. So these are some major contents that we are going to cover in this particular video lecture. First is the redox reaction, the introduction of it, what are the different electron carriers that are used in biochemical reactions in microorganisms, then the general overview of the metabolism, etc., that is the electron transport chain, and then the most important metabolic pathway in microbes, that is the oxidative phosphorylation, and we will end with the ATP synthesis, how the ATP synthesis is done with the help of enzyme ATP synthesis. So also please make a note that this particular section, that is the microbial metabolism part 1, is not having any questions from the previous year question paper. Okay, rest of the parts, that is the first, second, third as well as the fourth part, they are having in detail biochemical reactions. In those parts you will see that there are questions from previous year question papers. So let's start with the basic concept, that is the metabolism. So metabolism, we all know that it refers to some of the biochemical reactions which required for the energy generation. So the basic what we can say, the basic model of the or the basic motive of the metabolism is the energy generation, and then the use of this energy to synthesize the cellular material. So on right hand side you will see one figure which represents the overall metabolism, that is the catabolism and anabolism. So we all know that anabolism is nothing but the building of the blocks or making of macromolecules, whereas with the help of ATP, of course. Whereas if you see what exactly the catabolism is basically breaking down of the energy as well as converting larger molecules into precursor, and then these precursor molecules are again used in anabolism. So the ATP which is formed with the help of catabolism is again used in the anabolism. So there is a continuous energy flow as well as the continuous flow of the precursor molecules, okay, in between catabolism and anabolism. So metabolism is nothing but a change, okay, and the microbial metabolism is nothing but all chemical reactions occurring in a microbe during its growth. Okay, so we all know the growth cycle, the lag phase, the log phase, the stationary phase, and the death phase. Throughout this phases of the microbial growth, you will see there are multiple reactions or the multiple metabolic pathways that are taking place, okay, which are continuously involved in breaking down of the larger molecules, okay, that is the catabolism. Then will be the energy generation. Then again there will be anabolism, for example protein synthesis, peptidoglycan synthesis, okay, there are complex lipids which are formed, genetic materials are formed. All these are anabolic process which makes use of the precursor molecules which are formed in the catabolism, okay, so on and so forth. Then the next topic is the enzymes. Okay, so enzymes you all know are the biological catalyst. So catalyst, what is catalyst? Catalyst is any chemical agent or any, we can say, biochemical agent that speeds up the reaction. Okay, so there are different parts of this enzyme. Okay, so if you will say bioenzyme, so it has different parts such as it has a metallic cofactor, it has an apoenzyme part, it has a coenzyme part, so on and so forth. So although some enzyme consist entirely of proteins, most of them are consist of a protein portion, that is the apoenzyme. So apoenzyme is the protein portion, and then they have a non-protein component which we called as a cofactor. Okay, then ions of iron, zinc, magnesium, calcium, for example, they act as cofactor. If cofactor is an organic molecule, then it is called as a coenzyme. Okay, if the cofactor, that is for example iron, zinc, so these are the ions. Okay, but if the cofactor is an organic molecule, it will be called as coenzyme. Then apoenzyme is what? Apoenzyme is generally the inactive. So it is a inactive part. Okay. When it gets activated when coenzyme binds to it or any cofactor binds to it. So togetherly, apoenzyme and coenzyme or apoenzyme and cofactor forms a holoenzyme or a whole active enzyme, you can say. You can see on the right hand side the diagram. So this coenzyme and this apoenzyme, it togetherly makes what? Apoenzyme or the holoenzyme or the whole enzyme. Then moving forward, we have what exactly the metabolic pathways are. So metabolic pathways are nothing but anabolism and catabolism. So apart from anabolism and catabolism, redox reaction also plays a very important role in any of the microbial process or any of the biochemical reaction. So redox reaction is nothing but the reduction and oxidation reaction. In detail of it, we will see when we will study in detail about electron transport chain. Okay. Coming towards the next important part, that is the uh, energy or the electron carriers. So there are different electron carriers. So electron carriers are what? Electron carriers are nothing but the, we can say, these are the coenzymes in the cellular metabolism, such as nicotinamide adenine dinucleotide, that is NAD, and nicotinamide adenine dinucleotide phosphate, that is NADP. So what they do on right hand side, you can see. So whenever any in any biochemical reaction, if the electrons are released, these are the ones which will carry the electron just like a carrier, and they will provide this electron to the other biochemical pathways wherever it is necessary. See, in this reaction you can see the NADPH, so nicotinamide adenine dinucleotide phosphate, it has accepted one electron from some biochemical reaction and then it is providing it to a Kelvin cycle. So these are nothing but the electron carriers. So we have different electron carriers such as NAD, NADP, FAD, FMN. Then coenzyme Q, we have different cytochromes and the Redox. So we all know that the energy in the cells, it is stored as ATP. Okay, so it is stored in the form of ATP, adenosine triphosphate. This ATP is continuously, switching, or it is it's a cycle. Okay, just like we saw that in the first diagram itself, that is the anabolism and catabolism. Anabolism is what? Utilization of the energy. Catabolism is what? Breaking down of the larger molecules into smaller precursor molecules, which will be further used in anabolism. Plus, there will be a formation of ATP. So, anabolism and catabolism togetherly will, there will be a continuous flow of energy throughout the biochemical processes. So exergonic reaction, endogonic reaction are nothing but, so catabolism, what we can say, catabolism is one, these are the exergonic reactions which generates ATP, and anabolic processes which makes use of this precursor molecule and they form larger molecules, like, or the larger macromolecules, you can say, like proteins, vitamins, minerals, or the peptidoglycan. So these are the endogonic reactions. Why? Because they are utilizing this energy in the form of ATP. Now coming towards the most important part of the entire biochemical pathway, that is the electron transport chain. So I will be covering in very short what exactly the electron transport chain. In further video lectures, we will see in detail what exactly the electron transport chain is. Okay, so this is the overview of the electron transport chain. So back, ETC consists of series of electron carriers. We just now saw what the electron carriers are. So electron carriers are the molecules, or we can say the coenzymes that carries electron. Okay, and they transfer this electron from one molecule to another. Okay, they will carry this electron and then they will transfer. So that just like a carrier. So ETC consists of series of electron carriers that sequentially transfers the electron, and this depends upon their electrode potential, or you can say the capacity or their ability to retain electrons. Okay, retain as well as to accept electron. So electron flow generally begins from an electron donor to the strongest electron acceptor. For example, during glycolysis and TCA cycle, organic molecules, they donate electron to NAD plus molecules, converting them to NADH. So they basically reduce the NAD plus, converting it into NADH. Okay, and these NADH molecules, they act as electron donors. Okay, carriers as well as donors. So they carry these electrons from glycolysis as well as from the TCA cycle, and they donate these electrons in the electron transport chain. Okay, so a typical bacterial ETC, if you will see, typical bacterial electron transport chain, which is used in the process of respiration, it consists of what? It consists of basically three components. First is the substrate oxidoreductase. So there will be oxidoreductase, which will carry out the entire oxidation reduction. Then we have a quinone pool. Okay, so we have a quinone pool. Okay, and then we have a terminal electron acceptor. So this terminal electron acceptor, or we can say terminal oxidase complex, basically the electron acceptor. So this will change from one microbe to another. Okay, there are some microorganisms in which the electron acceptor, or the terminal electron acceptor, is different from the another microbe. Okay, that depends upon species to species. So ETC we will see in detail in upcoming lectures. Now coming towards the next part, that is the fermentation. Okay, so bacterial fermentation, we all know that it is a set of anabolic again and catabolic reactions that produces ATP at substrate level phosphorylation. Okay, so it produces ATP at, as I said, at substrate level phosphorylation. So what it is written, that pyruvic acid from glycolysis can itself become the electron acceptor. Then pyruvic acid can also be enzymatically altered and then can be saved, or it can serve as electron acceptor. Okay, so pyruvic acid, which is formed in the process of glycolysis, it acts as an electron acceptor in case during the fermentation process. Then we have NADs that are recycled again and they re-enter into glycolysis. The organic molecules that become reduced in their role as electron acceptor are extremely varied, and they are often useful products such as ethyl alcohol, lactic acid, propionic acid, butanol, etc. Okay, so what are these? So lactic acid bacteria produces lactic acid. Then there are some yeast which produces ethyl alcohol with the help of fermentation. We have propionic acid as well. We have butanol as well. Okay, so these are some of the products that are, or we can say the byproducts that are formed as a result of fermentation. Now coming towards the basic concept of substrate level phosphorylation. So what substrate level phosphorylation is? So substrate level phosphorylation basically refers to the formation of ATP from ADP and a phosphorylated intermediate rather than from ADP and inorganic phosphate. Okay, so there will be a formation of ATP, but from ADP and a phosphorylated intermediate and not the inorganic phosphate, which is the usual process. Okay, so that's what the substrate level phosphorylation is. So substrate level phosphorylation in eukaryotes, or you can say yes, in eukaryotes it occurs in mitochondria, whereas in, if we will see in prokaryote, it occurs in cytoplasm. So it can occur under both circumstances, that is the aerobic as well as anaerobic condition, and it provides a quicker but less efficient source of ATP compared to the oxidative phosphorylation. That's what the fermentation is. Fermentation is the major part in industrial microbiology. Okay, because it microorganisms produces byproducts such as ethyl alcohol, then lactic acid, propionic acid, butanol, etc. Now coming towards the most important part of the entire microbial metabolism, that is the oxidative phosphorylation, or you can say the electron transport link phosphorylation or the terminal oxidation. So it is the metabolic pathway wherein the cells use enzymes to oxidize the nutrients, thereby releasing the chemical energy in order to produce ATP. Okay, so oxidation is taking place as well as phosphorylation, that is the ATP synthesis is taking place. Okay, in eukaryote this is generally takes place in mitochondria, and in prokaryote of course it is in cytosol, or if we can say in the cytoplasm. So almost all the aerobic organisms carry out oxidative phosphorylation, and this pathway is so persuasive because it releases more energy than the alternative fermentation. So in fermentation we saw that it is a quick producer of ATP, but it is not as good as we can say as oxidative phosphorylation. So it is making use of nutrients, okay, to produce the ATP. Okay, so oxidation of nutrients to produce the ATP, that is nothing but phosphorylation. Okay, now the last part, that is the ATP synthase. So ATP is synthesized by the enzyme F1-FO-ATP synthase or ATPase. Okay, so this enzyme is the smallest known molecular machine which couples the proton translocation through its membrane. Okay, in diagram you can see the proton transport in its membrane, okay, through its membrane embedded hydrophobic domain. So this entire domain, this is the hydrophobic domain. So there is a transfer of proton, then there is again this transfer of proton. During this transfer, this entire cycle is taking place. So during this transfer, there is a generation of ATP. from ADP and again the inorganic phosphate. Okay, not the organic. Okay, so it's a membrane embedded hydrophobic domain. So hydrophobic domain of ATP or ATPase or ATP synthase is F0. Okay, and then the hydrophilic head piece or the hydrophilic part is the F1 molecule. Okay, you can see in the diagram. So hydrophobic is F0, hydrophilic or the one which is in the cytoplasm is F1. Okay, so ATP synthase lies across a cellular membrane and forms an aperture that protons can come across through it. Okay, it has an aperture through which the proton can enter. Okay, when there are low concentration, when there is a concentration gradient, you can say. So this electrochemical gradient is generated by the electron transport chain and it allows the cells to store the energy in the form of ATP for the later use. Okay, for specifically for the synthesis of macromolecules, okay, such as proteins, then vitamins, then we have peptidoglycans, so on and so forth. We have genetic molecules such as DNA, RNA, so on and so forth. Okay, so ATP synthase is present in plant in thylakoid membrane and it is located therefore in chloroplast. In case of cyanobacteria, the case is different. It is located in cytoplasm. Okay, so energy is produced from the movement of proton back across the membrane and is used by the this this proton is used coming which is coming back into the cytoplasm is used by the ATP synthase to make the ATP from ADP and P again it is the inorganic phosphate. So there are, as I said in the second slide itself, that there are no previous year questions from previous year questions paper, particularly for this part. So because this part is just the basic introduction, in later parts we will see in much detail what microbial metabolism actually means. That is the cycle. There are different interesting microbial cycles or the biochemical pathways are there. So for this part, I will take your leave. So thank you for watching. If you have any doubt regarding this topic, please comment down below and also don't forget to subscribe to the channel. Thank you so much.